Okay, and welcome. I'm a little bit early, but I thought it'd be worth starting this now. So I've got a deadline to finish by 6 o'clock. So we are doing an exam paper walkthrough of the AQA AS paper 2017, paper 1. This was actually requested, so here am I trying to keep people happy. So let us kick off. Oh, probably worth saying that I'm going to show you what I would have written, what marks I would have got, because I didn't get every single mark in this paper. I'm going to explain what I would have learned from the mark scheme and how I read the questions. So hopefully going through the whole lot and being helpful. So question 1. How many nucleotides are shown in figure 1? Well, you might remember that this is a single nucleotide phosphate. It's a DNA molecule, so deoxyribose, and one of the 4 bases. So we've got 1, 2, 3, 4, 5, 6, 7, 8, which is the right answer. Name the bond labeled X. That is a phosphodiester bond. If you have any questions as we're going through, do put them up in the chat. I'll be keeping an eye on it so that I can answer any as I go along. Okay, so moving on to the next question. What are the functions of DNA helicase and DNA polymerase? DNA helicase, it breaks the hydrogen bonds. And if that's the description, notice that this is only about describing. It's not about explaining anything. Just basically describe what it does. DNA helicase breaks the hydrogen bonds. DNA polymerase forms phosphodiester bonds. It's a really classic misunderstanding that DNA polymerase reforms hydrogen bonds. It doesn't. So worth getting that straight in your mind. There's the mark scheme, examiner's comments, and I got both of the marks there. Notice you're allowed to use the abbreviation H bonds for hydrogen bonds. Worth knowing that. Adenosine triphosphate is a nucleotide derivative. Contrast the structures between ATP and a DNA nucleotide to give 2 differences. Now, here is the crucial thing. If you're asked to compare or contrast, contrast is saying obviously what is different, comparing is saying what is alike and what is different. Contrasting, you're talking about the differences. Here's the really key thing. You have got to pick a feature that is present in both the ATP molecule and a DNA nucleotide, but say what is different about them. So you're picking a feature and you're saying how that feature is different in the 2 examples. In this case, ATP nucleotide, ATP and a DNA nucleotide. Here is the ATP molecule. You have got, it is made up of ribose sugar. It is made up of an adenine base. And it's made up of 3 phosphate groups. The ribose and the adenine base together are called adenosine, which is why this is called adenosine triphosphate. Notice though that the base in ATP is adenine, it is not adenosine. Get that, kind of try and get that clear in your mind. So if we've got this structure in front of us, hopefully we can see that there are at least 2 major differences between them. First of all, the 2 obvious ones to me is that the sugar, the pentose sugar that is in RNA is ribose, whereas in DNA nucleotide, it would be deoxyribose. And the other most obvious thing to me looking at this diagram would be that a DNA nucleotide has only 1 phosphate group, whereas the ATP has 3 phosphate groups. So here are my answers. Notice that I take the feature, in the first instance, the sugar, and I say what the sugar is in ATP, what the sugar is in DNA. And I do the same with the second feature of the number of phosphate groups. The other obvious thing that you could have said is the difference in the bases. In ATP, the base will always be adenine, Whereas in DNA, it could be adenine, thymine, cytosine, or guanine. So, or you could have said, no, yeah, because you're talking about DNA, not RNA. So it wouldn't be uracil, because uracil is obviously only an RNA. So hence this comment here by the examiners. So I picked up those 2 marks. Question 2. Student investigated the effect of 3 types of disinfectant on the growth of lactobacillus bacteria. And here is a summary method of what they did. Boiled the agar, put 0.5 centimeters cubed onto each of the agar plates, left some agar plates as controls, but then added to other plates different concentrations of the disinfectants, as you can see in this table. So we've got lysol, propan-2-ol and ammonia with various concentrations and showing how many colonies are left after, how many is it, 2 days. So explain the purpose of boiling the agar. This happened before pouring the plates. And this is so that the agar is sterilized. This is the agar gel that the lactobacillus is going to be growing on. We're only interested in lactobacillus, so we certainly don't want any other bacteria in that agar. So we boil it to sterilize it so that only lactobacillus will grow on the agar, because we've killed off everything else. Why do we transfer the same volume of liquid culture onto each agar plate? We are in effect inoculating each agar plate with the same volume of bacteria. Well, that's so that each agar plate has the same number of lactobacillus bacteria on it. Now, here is the mark scheme, and here are the notes, and this is the mark that I got. I didn't get this second mark because, crucially, I didn't include this statement here, so that we could compare the plates. Now, that's an important thing, because here we are talking about a control variable, and control variables have to be kept the same so that different parts of the experiment can be compared. So here we go. 3 disinfectants used by the student are these 3, which I've just mentioned. The liquid culture the student transferred was diluted 1 in 10,000. So for each centimeter cubed of original liquid culture, another 10 or 9,999 centimeters cubed of bacteria-free culture were added to it, diluting it 1 in 10,000. Use the information in this question, in this table, to calculate how many bacteria were present in 1 centimeter cubed of undiluted liquid culture. Well, this table tells us that in the circumstance where no disinfectant was added to the agar, 300 bacteria grew from 0.5, here's the important bit, 0.5 centimeters cubed of diluted liquid culture. So if we got 300 from 0.5 centimeters cubed of diluted culture, does it make sense that we have got to times 300 by 10,000, but then also by the factor, because we're being asked to find out how much is in 1 centimeter cubed of undiluted liquid culture, not half a centimeter cubed. We've got to times it by the factor of 1 over 0.5, i.e. 2. And therefore, the number is 6 million. Either of these answers would do, as you can see from the mark scheme, and you got a mark for getting halfway there. Okay. So the student concluded that the minimum concentration of propan-2-ol needed to stop the growth of the lactobacillus was 15 units. From this column of the table here and this bit here, because at 15 arbitrary units of propan-2-ol, no lactobacillus grew. This conclusion was incorrect. describe how you would obtain a more accurate estimate of the minimum concentration of propan-2-ol. And hopefully you'll realise that at 10 arbitrary units of propan-2-ol, 195 colonies grew. That's quite a lot of colonies when we started off with 300. And then if you jump from 10 to 15, you get 0. Well, that kind of tells me that somewhere in the middle of 10 and 15 arbitrary units, we're going to have a decreasing number of bacterial colonies growing. And quite possibly at some value between 10 and 15, 0 is going to be hit. And therefore, doesn't it make sense that we have got to carry out further trials between 10 and 15 arbitrary units? I could have said, e.g., 11, 12, 13 and 14 AUs. And then we've got to repeat those trials. We can't just do a one-off test to get our result. So, and that is where I got the 2 marks. Okie dokie. Question 3. Oh, this isn't very nice, or it doesn't look very nice. This is all about the digestion and absorption of lipids, which I have to say is one of the trickier parts of the AS syllabus. We have got this diagram here, which is showing the lumen, the hole in the middle of the small intestine, with lipid droplets in it, which are then seemingly broken down into fatty acids and glycerol. They are turned into micelles, and these micelles cross the wall of the intestinal epithelial cell, and they are somehow reformed into triglycerides in a vesicle, which then goes through this organelle here, and out the other side comes a chylomicron, which contains protein. And then the contents of the chylomicron is absorbed into the lymph capillary. First question, tick the box by the name of the process by which fatty acids and glycerol enter the intestinal epithelial cell. Well, there is going to be a higher concentration in the lumen where the lipids are being digested than there is in the epithelial cell. So it is definitely not active transport. It is also not osmosis, because osmosis only deals with the movement of water. We're now down to diffusion and endocytosis. Yes, endocytosis. Sorry, it's covered up on my screen. So we have got to decide between these two. Because, now here's the real crucial thing. This is remembering that anything that is lipid soluble will be able to pass straight through the phospholipid bilayer component of any cell membrane. Because if something is lipid soluble, it will dissolve in the phospholipid bilayer and be able to pass straight through. In this case, because the higher concentration is here, by simple diffusion. I'm going to go with diffusion. It isn't endocytosis because the micelles do not fuse with the cell membrane. They're not made up of quite the same thing. They're not made up of phospholipids, and therefore it isn't endocytosis. So it is diffusion. Explain the advantages of liquid lipid droplets and micelle formulation. Here we are, we've got to explain. This means our answers have got to have the word because in them, or so that, or as a result of, because that is, anything following those words is very, very likely to be an explanation. And personally, I would split my answer up into 2 sections, talking about lipid droplets and then talking about micelles. Now, lipid droplets to me is the kind of easier bit, and here is what I wrote. The formation of small lipid droplets greatly increases the surface area on which lipase enzymes can work, because the lipase enzymes can only work on the surface areas. So the smaller these lipid droplets get, the greater the surface area to volume ratio there is for the lipases to work on. And so that's what I put for that. And then I just said micelle formation allows for faster absorption, because theoretically fatty acids and glycerol, certainly the fatty acids, would go straight through the phospholipid bilayer. It allows for faster absorption into the epithelial cell. Wasn't really sure what they were looking for there, if I'm honest. So here is the mark scheme, and here are the examiner's comments. Notice I got the first mark point, got the first mark point here. And this is where I need to make a note in my revision notes, or really make a note in my brain. I absolutely knew mark point 2. And I also should have recognized that this question is about what makes the lipase digestion faster. And as I was writing this first bit, I nearly extended this sentence to say breaking down, hydrolyzing triglycerides into fatty acids and glycerol. But I didn't. I lost the mark here. And I didn't get the mark here because this English, the words that I used here, don't quite nail what they were looking for in this answer here. So for safety's sake, I gave myself 1 out of 3. Name structure Q and suggest how it's involved in the absorption of lipids. Structure Q, absolutely the Golgi apparatus. In any diagram, the Golgi apparatus looks like a kind of rounded stack of plates. That's the thing to look for. And the standard thing that the Golgi apparatus does is modify substances and packages them up. That is nearly always your go-to phrase about the Golgi apparatus. But here am I noticing that this is 4 marks. So I'm kind of going, okay, I might have got 2 marks by saying Golgi apparatus modifies and packages. But now I am going to use the diagram to really nail what it is I can write in terms of the detail that relates my standard answer to the question that is being asked. So I look at this diagram carefully and notice that it is modifying and packaging triglycerides. That's what's going into the Golgi apparatus. And what comes out the other side is a chylomicron, which is why I've written what I have. Notice that I'm being inspired by the question. And when I say the question, I'm talking about every bit of information that the exam paper gives me. I'm using that to inspire my answer. Here's the mark scheme. And here are the examiner's notes. And so I get mark point 1, mark point 2, and I think mark point 3. Yeah, so yes, because I talk about it by adding the proteins. I didn't get mark point 4 because if I'm really honest, I don't totally associate the fact that the Golgi, even though this often happens, Golgi is always involved in the secretion of something out of the cell. That often happens, but not always. So my learning point here is to kind of go, do you know, possibly I could have extended my answer if I had been more inspired by the diagram. And I could have also asked myself, did I really think that I had written enough for 4 marks? That's another way of looking at it. You know, push yourself to write something for every mark. That was possibly my downfall there. Hello, lovely to see everybody in live stream land. Thank you for joining me, and I hope that this is useful for you. Question 4. Figure 3 shows the volume changes in the left ventricle of the human heart during 2 cardiac cycles, and the numbers represent heart valves opening and closing. So this is not a graph that you would necessarily be familiar with. So I looked at this graph and I kind of went, okay, this is the ventricle filling up because the volume is increasing. The ventricles are filling. Here, the ventricles are decreasing in their volume really quite markedly. This is when the ventricles are contracting, with the blood exiting the heart through the arteries. And this is obviously specifically referring to the left ventricle and therefore the blood exiting through the aorta. Now, this is the graph that you would be more familiar with following your cardiac cycle teaching. And so now I'm going back to my knowledge of this graph to kind of go, okay, you know, what order do all of these valves close and open in? So if I'm remembering this graph, I kind of remember that, you know, the first thing that happens is that the grey line of the left ventricle increases, goes above the solid black line of the atrium. And therefore, if the pressure in the left ventricle is greater than the pressure in the atrium, those atrioventricular valves are going to snap shut. So here is where the AV valve closes, and obviously when the pressure of the left ventricle decreases, the AV valve opens. Similarly, when the pressure in the left ventricle goes up above the pressure in the aorta, the semilunar valves open, because the semilunar valves are orientated like this. So when the pressure is greater here than here, they get pushed back against the wall of the aorta. And similarly, the semilunar valves close when the pressure drops below that is in the aorta. So now I'm kind of going, okay, now I think I know which order this is that it is going in. So first of all, the first one is the atrioventricular valve closing. The second is the semilunar valve opening. The third is the semilunar valve closing, and the fourth is the atrioventricular valve closing. Not many students in 2017 got those marks. So there's the mark scheme. You got a mark per column, and that's how the marking worked out. Use figure 3 to calculate the volume of blood pumped per minute by the left ventricle. Now, just thinking about this logically, you are trying to work out from that graph the volume of blood that is pumped out of the left ventricle in 1 minute. Now, logically, doesn't it make sense that that requires you to know how much the volume of the left ventricle is and how many times the left ventricle contracts in order to empty the volume of blood out of it? So for those two pieces of data, we need to know the heart rate of the heart, and we need to know the volume of the left ventricle, because then, as you may remember, cardiac output equals heart rate times by the ventricular volume, cardiac volume. So in order to get the heart rate, we have got to work out how long one single heartbeat lasts. And so from this graph, I can work out, so if I, kind of go from here, I'm looking for when the pattern repeats itself. And can you see that if we take 0 as our starting point, the pattern repeats itself here? Notice that 5 little squares on the graph equals 0.2 seconds. So 1 little square equals 0.4. Therefore, the time for one heartbeat is 0.76. That tells us how long one heartbeat takes. But of course, we need to know how many of those heartbeats fit into a minute. So does it make sense that we have got to do 60 seconds, it's 1 minute, divided by the time it took for one heartbeat, about 3/4 of a second, and that equals 79 beats per minute. 79, we've got our heart rate. Now we need the cardiac volume, the volume of the left ventricle. Well, gloriously, this graph is volume of the left ventricle. Woohoo! So does it make sense that we need to get the volume of the left ventricle by taking the maximum volume, taking the minimum volume away from the maximum volume? So here is the maximum volume. Oh, I'm actually going to be oh yeah, from here, from here down to here. So 116 minus 40, which equals 76 centimeters cubed. So we can now times 79 by 76 to give us 6,004 centimeters cubed, which is just over 6 litres. And that sounds about right. Our heart beats 6 litres of blood around our body. If we've got a heart rate and a cardiac volume the same as this, you know, it's about 5-6 litres every minute. And here is the acceptable range, depending on exactly how you calculated those two figures. So that is your 2 marks. Explain the role of the heart in the formation of the tissue fluid. Formation of the tissue fluid. How the tissue fluid is made at the arterial end of the capillary bed. Well, if you remember your tissue fluid lessons, the tissue fluid is made as a result primarily because of the hydrostatic pressure of the blood at the arterial, maybe I should say it this way, at the arterial end of the capillary bed. And so this is what I put. And what creates that hydrostatic pressure? Well, that's our heart beating, or more particularly, the force that is created by the ventricles contracting. So this is what I wrote. The contraction of the ventricles causes high hydrostatic pressure. This causes the filtration of the blood contents, and I put in brackets everything except red blood cells, white blood cells, and proteins, out of the capillaries. And I must admit, looking at this now, I feel like I should put at the arterial end of the capillary bed, but I didn't. Anyway, here's the mark scheme. Here's the examiner's notes. So notice that they really did want you to use those words, contraction of the ventricles, rather than just pumping of the heart beating, contraction of the heart. They wanted you to be specific. I could really easily see that a lot of students would lose a mark here, even though they fundamentally understood what was going on. This is about using the A-level terminology and really getting your head around the fact that the muscle contraction produces the force that creates the blood pressure as the blood exits the heart. So I got mark point 1, but I didn't give myself mark point 2. I looked at the examiner's report on this, and the examiner's report, they were very strong on the fact that actually they really wanted students to appreciate that it was the water, prime and dissolved substances, that is forced out of the blood. Whereas, I'm going to be honest, this is often what I teach, that actually it is, you know, it's a load of stuff going out of the blood, except for those big or insoluble things, the cells and the large proteins. I didn't give myself the mark. I would make a point on my revision cards that actually I need to talk about water and dissolved substances being filtered out and not using the phrase everything except white blood cells, red blood cells, and proteins, unless the question was specifically targeting that kind of information. Learning from the mark scheme, not just learning the mark scheme. Lymphedema is a swelling in the legs which may be caused by a blockage in the lymphatic system. Suggest how the blockage would cause lymphedema. Well, here you need to know what the role of the lymph is in the formation of tissue fluid, and the lymph acts as a drain of any excess tissue fluid in your tissues around those cells. So the blockage stops excess tissue fluid being drained away. Therefore, tissue fluid accumulates around the cells, causing swelling. That's what happens. So here's the mark scheme and the comments, and I got the mark. here am I trying to absolutely nail why what I have answered, what I have said, answers the question, kind of closing the loop of my explanation back round to what they have asked me about. It didn't get me a mark. Maybe I should have realised it's only 1 mark. I'd always have a tendency to do it at A level if I've got the time. Question 5. Scientists measured the mean amino acid concentration from white wines made organically and non-organically. What test would they have used? Well, back to good old food tests. This is the Biuret test. Notice it's not the Benedict test, even though they both begin with B. Benedict is the carbohydrates, Biuret is the protein. Turn purple to give you a positive result. So notice all the other ways that you could describe this. Notice that they would give you practically any spelling of Biuret, except they would not allow Buret, because that is a piece of chemistry equipment, and they won't allow Beirut, not surprisingly either. Here you are given this particular amino acid, isoleucine. Don't need to know their names, you're just given this as an example. Circle the box around the part of the molecule that would be the same in all amino acids. So think of the name, amino acids. Needs to have an amino group in it, it needs to have an acid group in it, and it needs to not have the R group in it. So here is the circling, because this bit is what would be in every single amino acid. Amine group, carboxylic acid group, hydrogen around a central carbon, and it's then whatever is off that 4th bond of the central carbon, which will be the R group. That's the bit we're excluding. So we've got that and we've got the mark. Name the chemical element found in all amino acids that is not found in triglycerides. Well, that is nitrogen, because the only other elements are carbon, hydrogen, oxygen, and those elements are found in both triglycerides, in fact, all lipids, and in carbohydrates. Notice, though, that this says name. Don't ever use abbreviations when you are told to name something. I would never do so. And interestingly, they don't allow the chemical symbol as a substitute. Reasonably, always use the full name if you are asked to name something. Okay, scientists used a statistical test. They obtained a p-value of 0.04. What is the statistical test? Oh, sorry, I should have read that more carefully. That whether there was a significant difference in the amino acid concentration in the 2 types of wine. They've got a value for organic and they've got a value for inorganic. Can I suggest that those would be mean values? Hopefully that makes sense. And as a direct result of that, the statistical test is the student t-test, because that is the only statistical test that you know about that analyzes the difference between 2 means. Each statistical test can only be used on particular types of data, and that's the t-test one. Explanation of the test result. Well, you've been told that they've got a p-value of 0.04. Michael, I'm going to come to you. I've just seen your question. I'll come to you in a second. They've got a p-value of 0.04. Well, that p-value, p equals the probability that the number that they got out of the test was due to chance. That is 0.04 or 4%. The cutoff that biologists use is 5%, or p equals 0.05. If the probability is less than 5% that the result is due to chance, biologists will kind of go, right, we'll take that seriously. We'll say that the difference is significant. So difference between the 2 means is significant as p equals 0.04 and is less than 0.05, just stating the obvious, kind of. So I get all 3 marks there. Right, Michael, Carol, does one contraction heartbeat of the left ventricle ensure blood reaches all of the body, or is it the continuation of the heartbeat that makes sure it gets to where it needs to be? It is the contraction of the heart with one heartbeat that provides the pressure to get the blood out of your aorta, down to the tips of your toes and up to the top of your head. Quite remarkable. So there are two things that help your get the blood get to where they need to get to as a result of one heartbeat. That, and also sometimes the constriction of the blood vessels, because if the blood vessels, in particular the arterioles, constrict, the blood pressure increases, which allows it kind of to go a little bit further. If you imagine it's being, you know, almost pushed a little bit harder. I hope that makes sense. Fundamentally though, Michael, it is the single heartbeat that gets the blood from your heart to the extremities of your body. Hope that helps. Okay, moving on. I know I'm on a little bit of a mission, but I really want to try and get through this by 6 o'clock because I'm tutoring them. Okay, so question 6. We have the life cycle of a moss plant, which you know nothing about, and you do not need to know anything about. And it is late, and you are told that only the stalk and the spore capsule are diploid. And all the cells in all other stages are haploid. All the rest of this is haploid. This requires you to know the difference between haploid and diploid. Haploid is when the cell has only one copy of each chromosome in it, like in our gametes, in the eggs and in the sperm. Diploid is when in every cell there are 2 copies of each chromosome. In our body cells are diploid because we have the chromosomes in pairs. One has come from our father and one has come from our mother. So here we go. Which letter, A, B, C, or D, shows where meiosis occurs? Right, here is the key. Meiosis always reduces the number of chromosomes from diploid to haploid. So we are looking for the stage where that happens. And that is here because we are told that everything else is haploid. I couldn't care less that, see, everything else is haploid all the way around here. The number of cells are increasing, but that is mitosis. Mitosis can happen in haploid cells, but going from diploid to haploid, that is meiosis. So the answer is D, and there's the mark. Explain how the chromosome number is halved during meiosis. Okay, we have got to remember that at the beginning of, in fact, before either meiosis or mitosis, we have got the chromosomes have duplicated, haven't they? They've become those X-shaped chromosomes, sister chromatids that are stuck together with a centromere because duplication of the DNA has already happened. So each one of those X-shaped structures is one chromosome. So how do the number of chromosomes halve? Well, hopefully, logically, it makes sense that that happens when, during the stage of meiosis, when the homologous chromosomes, those pairs of chromosomes, one of those X-shaped chromosomes being the maternal one being the paternal one, they line up in the middle of the cell and then they are separated. One chromosome goes into one daughter cell and the other chromosome goes into the other. So that is what I have written. When homologous chromosomes line up in pairs and are separated, and it happens during meiosis 1. Independent assortment goes on, but that is irrelevant to this question. So I'm not even going to talk about it. They've just asked me how the number halves, not anything to do about variety. So I'm not going to go off on one about that. And, you know, each one goes into the daughter cells. And gloriously, that is actually the answer. So that's good. Michael, so would CPR produce the same effect? Yes, that is absolutely the plan. Yes, completely. If somebody's if somebody's heart stops, that is the whole aim of CPR, is to artificially keep that blood going around the body. Even if it isn't happening quite as quickly, you know, you might not get the rate quite right, but it's to keep that blood going around the body, to keep the oxygen being delivered to all the cells, to obviously keep them alive. Hope that helps. Here is a cell from the moss plant. It's in the second meiotic division. Oh, look, the 2 chromatids of each chromosome. Remember, we just had those homologous chromosomes separating into those 2 cells at the end of meiosis 1. And now we've got the chromatids of each chromosome being separated down into those final, as it were, 4 cells that are going to be produced as a result of meiosis. And so the question is, what is the haploid number of the chromosomes for this species of moss? Well, the haploid number, let's just say, you know, it's what ends up in those gamete cells. So it's going to be this one, this one, this one, this one, this one, and this one end up in one cell, there's 6, and the other 6 end up in the other cell. So the haploid number is 6. Crossing over greatly increases genetic diversity in this species. And can I just say, it does in any other organism that sexually reproduces. Describe the process of crossing over and explain how it increases genetic diversity. 4 marks. Not quite sure how the marks are separated out. You could kind of go, it's likely to be 2 and 2. Notice, because you are asked to describe and explain. Always keep an eye on the command words so that you know that you are actually answering what the question is asking. So I'm going to split my answer in half. I'm going to describe crossing over, and then I'm going to attack the explain bit. So here is my answer. And this bit ends up being the describe, and this bit ends up being the explain. Best I can do. Crossing over occurs when homologous chromosomes line up alongside each other, and non-sister chromatids, because we've got 1 chromatid from the paternal chromosome, obviously X-shaped, 1 chromatid from the maternal chromosome, they wind around each other, and then they exchange portions of the chromatids. That's the best I could do with regards to describing it. Explaining it, why does it happen, or what does it do? This produces new combinations of alleles in each chromosome, and then I go on to explain. Therefore, you get unique allele combinations in the offspring, equals genetic diversity. Each individual genetically unique, different alleles. Notice, same genes, different alleles. So here's the mark scheme, here's the notes, and here are where I got the marks. Mark point 1. They associate, they line up alongside each other. Chiasmata form, I didn't use that term, but I described it over here. Lengths didn't have to say equal. Lengths of non-sister chromatids are exchanged. I got that mark point. Combinations of alleles, I got that mark point. So, question 7, how are we doing? Okay, let's keep going. Any more questions? No, we're good. Question 7, describe how you would use cell fractionation. Hey, we're a completely different part of the syllabus. To obtain a sample of chloroplasts from leaf tissue. Do not include in your answer information about any solutions. In other words, they are not interested in why it is isotonic, buffered, and chilled. Yeah, forget all of that stuff, much as you'd love to put it in. Right, so here am I. This is what I put. First of all, you've got homogenize. I knew that was going to be a term. I can pretty much visualize what is going to be on the mark scheme here. Homogenize the leaf tissue using a kitchen blender, for example. It's generally what schools end up doing. Filter through gauze to produce the homogenate. The filtering removes all the cell debris. Spin the homogenate in a centrifuge to separate out the nuclei from the supernatant. Notice I don't need to know any spin speeds, all right? But the standard answer is that the first spin takes out the nuclei, and then you respin the supernatant, the liquid on top of the pellet, and you respin that at a higher speed to cause the pellet to contain the chloroplasts. And then you separate that from the supernatant. So there's the mark scheme, and I pretty much knew what was going to be on it. Wasn't, I added a bit about the whole filtering thing, but you're never quite sure. So, and that is where I got the marks. Table 3 shows the features of a mitochondrion and a chloroplast. Tick to complete. So, double outer membrane. Both mitochondria and chloroplasts have a double outer membrane. They have starch grains. The only cells that will have starch grains in them ever will be plant cells, and they're often found in the chloroplasts, never in a mitochondrion. Diffusion of oxygen into the organelle, mitochondrion, never chloroplasts. Chloroplasts only use carbon dioxide to make the glucose. So there's the mark scheme, and you've got a mark for each correct row. So whoopie-doo, I got the 3 marks. Give a function of a mitochondrion. Well, aerobic respiration. You're going to learn about that with bells on in year 13, but at AS level, you should know that it is aerobic respiration. To release energy, you can never ever say that energy is produced or made, because it is not a chemical thing. And thermodynamics laws say that energy can neither be made nor destroyed. So you cannot use those words. But you could say, I've put in the form of ATP, you could say producing ATP, because ATP is a chemical, it's a thing. So you can use the word produce there. So here we go. So I got the mark. Right, we're looking at the effect of exercise in these two. So they do a training program, and they test to see the number of mitochondria in the muscle before and after the training. And they also test to see the mean area of the inner mitochondrial membrane before and after the training. And here's the really key thing. Here's the clue in the question. Here is the standard deviation bars. What do the data suggest about the effect of the exercise program on mitochondria? Well, figure 7 tells us, because these standard deviation bars overlap, it says that there is no significant difference between the number of mitochondria before and after training. However, because these standard deviation bars don't overlap, that tells us that the difference in the area of the inner mitochondrial membrane is a significant difference before and after training. And importantly, that difference, that change, is that it increases. Here's my little tip to you. If you ever want to write the word change or has an effect, try and think, can I actually use the word increase or decrease? Because if you can, use it, because that is often where the mark is. So here we go. Oh, that's the rest of my answer. Here is the mark scheme. So I ended up getting both of the marks because I spotted it's an absolute classic. If you see standard deviation bars, you can be guaranteed that you should have some reference to them in your answer at some point in the question. And if you haven't, you know you've missed something. Question 8, give 3 properties of water that are important in biology. Oh, what to choose, what to choose. Here we go. Universal solvent. Dissolves everything because it itself is a polar molecule, so it dissolves everything that is either polar or ionic. Ions. High specific heat capacity and a high latent heat of vaporization. Notice it doesn't say state and explain. It just says give. Just means state, just say what they are. Don't have to do anything else. Mark scheme, so you've got a whole load of possibilities. Here is the examiner's comment, fair enough. And so I got those marks. Classic osmosis experiments. Potato chips put in different concentrations of sucrose for, I think, 24 hours, weighed before, weighed after. Hey ho, there's a mass change. So, student produced sucrose solutions with different concentrations. Name from a concentrated sucrose solution. What's the method? Well, it is dilution series. That was the answer. Serial dilutions were also, it was also accepted. A serial dilution strictly is when the dilution is the same factor every time. So if it was being diluted by a tenth every time, the dilution series would go down, for example, from 1 to 0.1 tenth, 0.01 a tenth of that, 0.01 tenth of that. Yeah. Here, it is not an equal factor of dilution. The intervals are equal, it isn't an equal. So this is strictly a dilution series, not a serial dilution, but clearly let's not get too hung up about it. Anyway, maybe that was just too much information, apologies. Calculate the ratio of final mass to initial mass of the potato chips. Okay, all right. And plot a suitable graph of your process data. Process data just means use the bits of data that you've just calculated. Express the ratios as a single number. This is actually trying to help you. For example, 5.26 to 1, final mass to initial mass would just be expressed 5.26. And this is actually a standard scientific convention, because if you express a ratio, the second number should always be 1 so that you can compare different ratios. Very hard to compare 4 to 1 and 9 to 3. Not quite sure, yeah? So, but if you made the second number 1, 4 to 1, 9 to 3 is 3 to 1, yeah? You can suddenly see which one is the larger ratio, because if the convention is the second number is always 1, you can then compare them. So final to initial. So here's the thing: how do you make that last number, the initial number, 1? Well, you divide it by itself to make this 1, and then of course you've got to divide the final mass by the initial value as well. So fundamentally, all you're doing here is dividing the final mass by the initial mass to make these numbers, to get these numbers. All of these strictly, well, I could have written 1.37 to 1, but they've actually said, drop the 1, if you hope that makes sense. So, and hopefully, just by looking at the raw data, you can see here we've got an increase in mass above 1. Hopefully that makes sense. And as the concentrations of sucrose increase, the amount of mass loss increases. Hopefully that makes sense too biologically. Here is my graph. Final to initial mass ratio. No units, it's just a number. Sucrose concentration along the x-axis because it is the independent variable. Here is the mark scheme. Here are the examiner's points. I get the first mark because I get the axes the right way round and the scales are linear. These intervals are the same each time. I get the labels right, and I plot correctly, and I join with a straight line. I could have used a smooth curve, Notice if you're plotting experimental data, you never extrapolate beyond what your data has told you. Okay, so explain the result of the chip in 0.8 moles per decimeter cubed of sucrose solution. 0.8, okay, this is 0.81. In other words, as we can see from the raw data, there has been a loss in mass. Why has there been a loss in mass? Well, that's because water has moved by osmosis from the potato cells into the sucrose solution down the water potential gradient, which is exactly what I wrote. Here's the mark scheme. Here's the examiner's comments. I only got one mark. So even though I said down the water potential gradient, this would be my standard answer. I'm going to be honest. Down the water potential gradient, which rather implies, to say the least, that here the inside of the cell has got the higher water potential, the sucrose solution has got the lower water potential. I still don't get the 2 marks. You absolutely had to say where specifically, you had to spell out where was the higher water potential and where was the lower water potential. At which point I'm kind of just putting my hands up and kind of going, you know, whatever. But equally then a little bit frustrated because it's not like I didn't know that. I would try and remember that for another similar osmosis question. But to be quite frank, in an exam, I'm not sure I'd ever write anything different. Number 9, okay, we may not get through all of this. Define the following terms. Here are the answers. Species, individuals can interbreed and produce fertile offspring. There's the key word, fertile. The offspring can themselves produce offspring. Species richness is the number of different species in the habitat. Okay, so I got the 2 marks here. We're given a load of, each dot here is a catch of fish. And it is plotted at the depth at which the catch was made and the species richness of that catch. How many different species of fish were there in the catch? And so you can see that, for example, you know, the depth increased, but oh my goodness, the species richness of the catches was, let's just say, fluctuated rather a lot. There doesn't seem to be a particular trend. First question is, what is the modal value of the species richness? The mode is the most frequent value. So we're looking to see which value of species richness occurs the most times. And it is here. It is 5. 68% of all the fish caught in the investigation come from sample A. This was a huge, this was a huge catch. All right. Student thought that this showed that sample A had a greater index of diversity than any of the other samples. It is not possible to draw this conclusion. Give reasons why. Well, the way my thinking went was, okay, what does the index of diversity require you to have? Even if you don't remember this flipping equation, which is pretty horrible, you might remember that it involves big N's and little n's. Little n's means that you need to have the number of individuals of each species recorded. And big N is the total number of individuals overall. So I wrote this because you do not have the following data. You don't have the number of individuals in each species, nor the total number of individuals. Obviously, the total comes from knowing this. And I also said that sample A could be all the same species. I'm just, arguing against why just because it's a big catch means it's got a lot of different species in it. Could be a shoal of herring. Anyway, here's the mark scheme. Here's the examiner's comments. I only got 1 mark out of 3, which was frankly disappointing. They would not allow you to say that sample A could be all the same species of fish. Personally, I find that really harsh. It is feasible that you could have just caught a shoal of a single species, but hey ho. So, and I only less than 0.25% of the students in 2017 got 3 marks. Hats off to them, is what I say. Right, question 10. Right, I am going to have to go now, I'm afraid. I will, what I will do is that if you're interested, I have a website, www.biologycarol.co.uk. This walkthrough of this exam will be on that website in completely edited form within the next week or so. That website has all the videos for the whole of the AQA and the OCRA syllabuses. Can I just say that means that it's going to have most of the content for every A-level specification. You can subscribe to those videos for a really, I hope you'll agree, reasonable amount of money for a year. If that would help you, you would have my teaching on tap, if that might be useful. Anyway, I am going to have to love you and leave you now. Thank you for joining me, and I will see you next week, where hopefully I will be going through, paper 2 of the AS paper. See you next week. Bye-bye.